For centuries, Chinese and European populations have lived thousands of miles apart, shaped by different climates, diets, migrations, and civilizations. Scientists always knew their DNA was different, but they didn't fully understand just how complex those differences really were. Then researchers began comparing thousands of genomes across East Asia and Europe using modern sequencing technology, and the results started challenging some of the assumptions scientists had held for generations. Genetic differences were appearing in metabolism, disease risk, drug response, and other biological systems. Yet beneath those differences was an even bigger surprise, an extraordinary amount of shared ancestry stretching deep into human prehistory. The more scientists compared the two populations, the more complicated the story became, and the results were forcing them to rethink what human genetic difference really means. For years, popular discussions about human genetics followed a remarkably simple narrative. Modern humans originated in Africa, expanded outward, and eventually separated into populations that adapted to different environments. Europeans developed lighter pigmentation in northern environments. Some East Asian populations developed distinctive physiological adaptations. Tibetans evolved extraordinary high-altitude biology. Populations living in different climates experienced different diets, diseases, and demographic histories. None of this is controversial. What became misleading was the assumption that these differences could be organized into a few clean biological boxes. Modern genetics has shown that human populations are not isolated genetic islands. They're overlapping populations connected by migration, interbreeding, and shared ancestry. Chinese and European populations are different enough that ancestry can often be inferred from genome-wide patterns, but they remain overwhelmingly similar at the level of basic human biology. The real scientific question is therefore not whether the populations differ. It is which differences are real, why they appeared, and what those differences can teach us about the human past and the medicine of the future. The first surprise begins with geography. Today, China and Europe appear separated by enormous distances and distinct civilizations. But those borders are extremely recent compared with human evolutionary history. Tens of thousands of years ago, there were no countries called China or Germany, no European Union, no passports, and no political boundaries stretching across Eurasia. Humans moved through forests, grasslands, river valleys, deserts, and mountain corridors. Some groups stayed relatively isolated for long periods. Others migrated hundreds or thousands of kilometers. Populations that had once been connected could separate, while distant groups could later meet again. The modern genomes of East Asians and Europeans are therefore the result of thousands of demographic events layered on top of one another. When scientists compare them today, they are not comparing two populations that independently invented themselves. They are comparing two branches that emerged from a much older network of human populations. The deepest connection between these populations reaches back to Africa. The ancestors of most people living outside Africa today descended from populations that expanded out of Africa tens of thousands of years ago. The exact timing, number of migration waves, and demographic details remain subjects of active research, but the central conclusion is robust. Modern Europeans and modern East Asians share ancestors who lived long before either population existed in its current form. This matters because physical appearance can create an exaggerated impression of genetic distance. Skin pigmentation, hair form, facial features, and other visible characteristics are noticeable precisely because they are visible, but they represent only a tiny portion of human biological variation. Beneath those traits, Chinese and European genomes contain the same fundamental biological machinery. The same genetic code builds cells, the same chromosomes carry genes. The same molecular systems repair DNA, produce energy, regulate immunity, and maintain tissues. Difference exists, but it exists inside a much larger framework of common ancestry. Ancient DNA has made this story even more complicated. One of the most revealing examples comes from Avar-period Cave in Bulgaria, where researchers sequenced genomes from early modern humans dating to roughly 45,000 years ago. These individuals were not simply the direct ancestors of modern Europeans. Their genomes showed stronger genetic affinity to later East Asian and indigenous American populations than to present-day Europeans. That discovery was extraordinary because it demonstrated that the first modern humans to enter Europe did not necessarily become the main ancestors of later European populations. Some early groups disappeared or contributed little ancestry to subsequent populations, while other groups arrived later and became more important. Human migration was therefore not a single wave moving west and then stopping. It was a dynamic process involving replacement, persistence, mixture, and disappearance. The modern genetic map of Europe is a later product of this complicated history, just as the genetic map of East Asia is. This discovery also changes how we think about the supposed split between Europeans and East Asians. It is tempting to imagine a single ancient population leaving Africa, reaching Eurasia, and then dividing neatly into a western branch and an eastern branch. Real genetic history is rarely that clean. Different groups could separate at different times. They could remain in contact after beginning to diverge. They could exchange genes long after geographic separation began. Some branches could disappear while related branches survived. The result is not a perfect fork in the road. It is more like a network of paths that repeatedly divide and reconnect. Ancient genomes allow researchers to identify some of those paths, while modern genomes preserve traces of others. That is why a 45,000-year-old individual from Europe can sometimes tell scientists something about the ancestry of populations living thousands of kilometers away in Asia today. Now move forward to the most important modern comparison. In 2025, researchers published a major cross-ancestry analysis examining the genetic architecture of metabolites in Han Chinese and European populations. The study began with 10,192 Han Chinese individuals and measured 171 metabolites using nuclear magnetic resonance-based metabolomics. Researchers identified 15 genetic associations with metabolites, and 8 were replicated in an independent Chinese population of 4,180 people. Then came the crucial comparison. The scientists integrated those Chinese data with genetic information from 213,397 people of European ancestry from the UK Biobank. Instead of simply finding that the two populations were different, the combined analysis identified 228 additional variant metabolite associations and improved the precision with which researchers could locate important genetic regions. In other words, comparing Chinese and European DNA actually made the biological picture clearer. The differences between ancestries became a scientific tool for discovering shared human biology. That finding is easy to underestimate. Imagine trying to understand a complicated machine while looking at only one version of it. You can identify many parts, but sometimes you cannot determine which component is actually responsible for a particular function, because several components vary together. Now imagine having a second version of the machine in which those components vary differently. Suddenly the relationships become easier to identify. This is one reason ancestry diversity matters in genetics. Different populations have different patterns of genetic variation and linkage. When researchers combine them carefully, they can sometimes narrow down the location of causal variants more effectively. Chinese and European genomes therefore do not simply compete with each other as biological datasets. They can complement one another. The comparison becomes valuable precisely because the populations share the same underlying human biology while carrying different frequencies of many genetic variants. The medical implications are enormous. Metabolites are chemical substances involved in processes throughout the body. They influence energy production, lipid transport, amino acid metabolism, inflammation, and many other biological systems. In the 2025 cross-ancestry study, researchers used statistical genetic methods to examine how metabolite levels relate to disease. Their analyses provided evidence that genetically predicted triglyceride levels in high-density lipoprotein were associated with coronary artery disease risk, while genetically predicted glycine levels were associated with heart failure risk in both ancestries. The significance of this is not that Chinese people have one metabolism and Europeans have another. It is that shared biological mechanisms can be discovered more precisely when researchers stop treating one ancestry as the universal reference. The human body operates through common pathways, but population-specific genetic variation can alter how those pathways behave across individuals. This leads directly to one of the biggest problems in modern genomics, the imbalance of genetic data sets. For many years, people of European ancestry were heavily overrepresented in genome-wide association studies. That imbalance did not mean scientists intentionally ignored everyone else, but it created a statistical problem. Genetic discoveries made in predominantly European data sets can perform less accurately when transferred to populations with different ancestry. A risk score trained in one population may not predict disease equally well in another because allele frequencies, linkage patterns, and environmental exposures differ. The answer is not to abandon European data sets. It is to build much larger data sets representing East Asian, African, South Asian, Indigenous, Oceanian, Latin American, and other populations. The future of precision medicine depends on understanding the full range of human genetic variation, rather than treating one population as the default. A major 2025 Nature study illustrates this movement toward population-specific prediction. Researchers developed polygenic risk scores specifically for people of Han Chinese ancestry using large-scale genomic and health data. The study emphasized a problem that has become increasingly clear. Most genetic studies have historically focused on European ancestry, creating an imbalance in the ability to predict complex disease risks globally. The researchers identified population-specific genetic variants and developed risk models for cardiometabolic disease, autoimmune disorders, cancers, infectious diseases, and other traits. Their findings were validated using independent data sets, including the Taiwan Biobank and East Asian participants in large international cohorts. This is not evidence that Chinese people have fundamentally different diseases. It is evidence that genetic prediction can become more accurate when the population being studied is actually represented in the data set, and that distinction is critical. A genetic variant can be common in one population and rare in another without creating a biological wall between the populations. Consider the ALDH2 variant that is substantially more common among East Asian populations. It affects the metabolism of acetaldehyde after alcohol consumption and can influence the physiological response to alcohol. This is a real population difference, but the underlying metabolic pathway exists in everyone. The variant simply occurs at different frequencies because of population history and evolutionary processes. Similar examples appear throughout pharmacogenomics. Variants affecting how people process medications can have different frequencies across populations, which means that the same drug can produce different distributions of responses. This is one reason modern medicine increasingly needs ancestry-diverse clinical research. The goal is not racialized medicine. The goal is individualized medicine. The same principle appears in genetic disease risk. A variant associated with a disease in European populations may not be the strongest predictor in East Asian populations. Sometimes the same gene is involved, but different variants are important. Sometimes the same variant exists in both groups, but at very different frequencies. Sometimes the apparent difference disappears after researchers account for environmental and socio-economic factors. And sometimes a cross-ancestry analysis reveals that a biological mechanism is shared even though the statistical signal looks different in each population. This complexity is why responsible genetic research avoids simple statements such as Chinese people have this gene or Europeans have that gene. Human genetics almost never works in such absolute categories. It works in probabilities, frequencies, interactions, and distributions. The history of diet provides another fascinating example. Human populations have experienced different food environments for thousands of years, and natural selection has sometimes acted on genes involved in digestion and metabolism. European populations provide a famous example through lactase persistence, the ability to continue digesting lactose efficiently into adulthood. This trait became common in several populations with long histories of dairying. East Asian populations developed other dietary histories, including long-standing agricultural systems in which grains and other carbohydrate-rich foods were important. Variants affecting starch digestion, lipid metabolism, and other pathways can therefore differ in frequency across populations. But again, adaptation is not a ranking system. It is a response to local conditions. A trait that was useful in one environment may have been neutral or less useful somewhere else. This is one reason the idea of genetic superiority makes no scientific sense. Evolution does not have a universal finish line. A population is not becoming better in an abstract global sense. It is becoming better adapted if selection is occurring to particular conditions. A gene that helps conserve energy might be useful during food scarcity, but disadvantageous in an environment with abundant calories. A variant that improves resistance to one pathogen might increase vulnerability to another. A trait that helps survival at high altitude may not matter at sea level. Evolution is full of trade-offs. It creates diversity because different environments create different challenges. Chinese and European populations are therefore not two contestants in a genetic competition. They are examples of how one species responds to different histories. High-altitude adaptation makes this especially clear. Tibetan populations have developed remarkable physiological adaptations that allow them to live at elevations where oxygen levels are much lower than at sea level. Some of the genetic architecture involved is connected to ancient Denisovan-related ancestry, including variants affecting oxygen regulation. But Tibetans are not simply more evolved than people living at sea level. Their physiology is adapted to a specific environment. A trait that is beneficial on the Tibetan plateau would not necessarily provide an advantage in London, Shanghai, or coastal Africa. The correct scientific language is adaptation, not superiority. This distinction becomes even more important when comparing broad populations such as Chinese and Europeans, because each contains enormous internal diversity. Han Chinese populations themselves are not genetically identical from north to south. Geography, ancient migration, historical population expansions, and interactions with neighboring groups have produced measurable regional differences. Northern Han populations have strong ancestry connections to ancient populations associated with northern China, while southern Han populations show additional genetic influence from populations farther south. These differences do not undermine the concept of Han identity. They demonstrate how a large population can expand across a vast geographic region while incorporating local ancestry. A similar pattern exists in Europe, where northern, southern, eastern, and western populations have different mixtures of ancient ancestry. The words Han Chinese and European therefore describe broad population histories, not single genetic profiles. Ancient DNA from China has helped reconstruct some of this history. Archaeological sites in the Yellow River region reveal long-term population continuity associated with early agricultural societies, but continuity does not mean isolation. ancient communities interacted with neighboring groups, and later migrations transformed the genetic landscape. Northern China became a major demographic center, from which populations expanded into surrounding regions. Southern China developed its own complex history involving ancient populations and later movements associated with agriculture and state expansion. The modern Han population is therefore the product of both continuity and movement. Its genetic history cannot be reduced to a single ancient tribe or one isolated origin. Europe tells the same kind of story from another direction. Ancient genomic studies have shown that present-day Europeans derive ancestry from multiple, deeply differentiated ancient populations, including Western hunter-gatherers, early farmers connected to Anatolia and the Near East, and populations associated with the ancient Eurasian steppe. Later migrations added further complexity. The modern European genome is therefore not a frozen continuation of one Stone Age population. It is a layered record of demographic change. This is why comparing modern Chinese and European DNA without ancient DNA can sometimes produce a misleadingly simple picture. The modern populations are endpoints of long journeys, and ancient genomes allow researchers to reconstruct the paths between them. The Eurasian steppe was especially important in connecting Eastern and Western populations. For thousands of years, people moved across grasslands, stretching from Eastern Europe toward Central and East Asia. These movements carried genes, technologies, languages, and cultural practices. Some migrations were large enough to dramatically change the ancestry of populations they encountered. Others involved smaller groups whose genetic contribution was limited but detectable. Modern populations can therefore contain ancestry from people who lived far outside the geographic region now associated with them. A genetic component that looks European today may ultimately trace back to an ancient population that lived in Central Eurasia. Likewise, ancestry found in East Asia may preserve signals of ancient movements that crossed regions now divided by modern borders. This is why ancestry should never be confused with nationality. A person's DNA does not carry a passport. It carries segments inherited from ancestors who lived at different times and places. Modern countries are recent compared with the evolutionary history of our species. Even ancient cultural identities do not map perfectly onto genetic populations. People can adopt languages, religions, or political identities without completely replacing their ancestry. Conversely, a population can disappear as a distinct cultural community while leaving a substantial genetic legacy in later populations. Genetics and culture interact, but they are not identical. The comparison between Chinese and European genomes also challenges another popular misconception, that visible physical differences must reflect equally large differences throughout the genome. They do not. A relatively small number of genetic regions can have large effects on visible traits such as pigmentation or hair characteristics. Natural selection can change these traits relatively quickly when environmental pressures are strong. But most of the genome remains shared. That is why two people can look noticeably different while being genetically extremely similar overall. Appearance is not a complete summary of ancestry. It is one visible outcome of a much larger biological system. Modern genomic technology can detect ancestry because the signal is distributed across thousands of genetic markers. A single variant rarely tells the whole story. Instead, researchers examine patterns across the genome. If many markers occur at frequencies associated with particular populations, statistical models can estimate ancestry, but those estimates depend on the reference populations included in the database. If the database lacks sufficient representation from a region, the algorithm may have difficulty distinguishing closely related ancestries. This is another reason recent Chinese genomic research is so important. Adding large East Asian datasets makes ancestry and health predictions more accurate for people whose genetic history was previously underrepresented. The 2025 Han Chinese Polygenic Risk Study demonstrates this directly. Researchers were able to identify population-specific risk variants and construct prediction models that performed well across independent datasets. Some of the strongest signals involved cardiometabolic diseases, autoimmune conditions, cancers, and infectious diseases. The important message is not that these diseases belong to Chinese people. They do not. The message is that the genetic architecture of complex diseases can differ in frequency and effect across populations, and medical prediction improves when those differences are represented. A global medical system cannot rely on one ancestry and expect perfect predictions for everyone. Longevity research provides another window into the same problem. Scientists have studied exceptionally old Han Chinese individuals to understand how genes and lifestyle interact in long life. In 2026, researchers analyzed 1,545 participants aged 80 and older from the China Hainan Centenarian Cohort Study. They constructed a polygenic score associated with longevity and combined it with a score representing 11 modifiable risk factors. The results showed that favorable lifestyle-related factors were strongly associated with lower mortality, while genetic predisposition to longer lifespan also had an independent association. Participants with both favorable modifiable factors and higher genetic predisposition had the lowest mortality risk. Most importantly, the study showed that unfavorable modifiable factors could substantially weaken the benefits associated with genetic predisposition. Genes matter, but they do not eliminate the importance of behavior and environment. That finding is important because genetic stories are often exaggerated. If scientists identify a population-associated longevity variant, headlines can make it sound as though a population has discovered a secret genetic formula for living forever. Reality is much more complicated. Longevity is influenced by many genes, health behaviors, medical care, socioeconomic conditions, environment, and chance. The 2026 Chinese cohort study demonstrates precisely that interaction. A favorable genetic profile was associated with lower mortality, but favorable modifiable factors were associated with an even larger reduction in risk. This is a powerful reminder that population genetics should not be interpreted as destiny. DNA influences probabilities. It does not dictate an individual's future. The same caution is essential when discussing the brain. The viral version of this story often claims the same caution is essential when discussing the brain. The viral version of this story often claims that Chinese and European populations have dramatic genetic differences in intelligence, memory, spatial reasoning, or brain development. That is not supported by the evidence presented in the major recent Chinese-European genomic studies. Genes involved in brain development exist in all human populations, and variants can differ in frequency. But that does not establish population-level differences in complex cognitive abilities. Traits such as intelligence are extraordinarily complex, influenced by thousands of genetic variants as well as nutrition, education, childhood environment, socioeconomic conditions, health, culture, and countless other factors. It is scientifically irresponsible to take a population frequency difference in one gene and turn it into a claim about the intellectual abilities of an entire population. This distinction matters because human genetics has a dark history. In the 19th and 20th centuries, researchers sometimes used biological differences to construct racial hierarchies. Those ideas were later incorporated into eugenics and discriminatory policies. Modern genomics does not support those conclusions. The existence of population structure is not evidence of superiority. Genetic variation is not a moral ranking. A population can have a higher frequency of a particular variant without being better or worse. It simply has a different history. The scientific challenge today is to study real variation without turning it into a hierarchy that the data cannot justify. There is also a major difference between population-level research and individual prediction. Suppose a genetic variant is twice as common in one population as another. That does not mean every individual in the first population has the variant, or that every individual in the second population lacks it. The distributions overlap. An individual Chinese person may have genetic variants more common in Europeans, while an individual European may carry variants more common in East Asia. Ancestry changes probabilities, not certainties. This is why personalized medicine ultimately needs individual genomic information rather than racial labels. Population ancestry can help researchers understand patterns, but the genome of the individual provides the more direct biological information. Another fascinating discovery comes from the study of shared ancestry itself. When researchers analyze enough genomes, they can find genetic segments inherited from common ancestors who lived surprisingly recently. The fact that Chinese and Europeans have different population histories does not mean their ancestors stopped interacting. Eurasia remained connected for thousands of years. Trade routes, migrations, warfare, and political expansion repeatedly moved people across long distances. Some historical episodes produced detectable ancestry contributions. Others left little trace. The genome, therefore, contains both ancient separation and later reconnection. The two processes happened simultaneously. This is why genetic history is better represented as a braided river than a simple tree. A tree suggests that once two branches split, they remain separate forever. A braided river allows channels to separate and reconnect. Human populations behaved more like the second model. Groups diverged, migrated, and sometimes mixed again. Some genetic branches became dominant, while others disappeared. A modern genome contains pieces of this complicated history. When scientists compare Chinese and European DNA, they are effectively reading two sections of the same braided river. The latest research also demonstrates that new discoveries can emerge when researchers stop analyzing populations separately. The 2025 metabolomics study began with Chinese data, replicated findings in another Chinese cohort, and then added European data. That cross-ancestry approach increased the number of detectable genetic associations and improved fine mapping precision. This is a powerful lesson. Diversity in research populations is not merely about fairness. Although fairness matters, it improves the science itself. Different ancestries provide different genetic variation, and that variation can help reveal causal mechanisms. A study that includes many populations can sometimes answer questions that no single population can answer alone. So what exactly shocked scientists when they compared Chinese and European DNA? Not a secret genetic superiority, not a hidden species boundary, not evidence that one population evolved separately from humanity. The real surprise is the combination of similarity and difference. Chinese and European populations have distinct demographic histories. They carry different frequencies of many genetic variants. Some variants influence metabolism, immunity, disease susceptibility, or drug response. Yet the biological systems controlled by those variants are deeply shared. And when scientists combine the populations, they often learn more about both. The discovery also changes the meaning of medical research. For decades, scientists asked questions such as, which genes influence cholesterol, or which variants increase diabetes risk? The next generation of research increasingly asks a more precise question. Which genes influence this trait across different ancestries? And do the same mechanisms operate in the same way? That shift is crucial. A variant may be causal in one population, but difficult to detect in another, because its frequency is different. Another variant may be common in one ancestry and rare elsewhere. By comparing multiple populations, scientists can separate universal biology from population-specific genetic architecture. This is already happening in cancer research, cardiovascular genetics, autoimmune disease, and pharmacogenomics. Large data sets containing people from different ancestries allow researchers to test whether genetic associations replicate. When a signal appears across populations, it becomes stronger evidence for a shared biological mechanism. When a signal differs, scientists can investigate why. Sometimes the answer is genetic, sometimes it is environmental, sometimes it reflects differences in healthcare access or lifestyle. Sometimes the original association was simply a statistical artifact. Cross-ancestry research therefore makes genetic science more skeptical and more accurate. And there is another important lesson hidden inside the comparison. Genetic diversity is not evenly distributed across the world. Africa contains the greatest human genetic diversity because modern humans have lived there for the longest continuous period. Populations outside Africa experienced founder effects and bottlenecks associated with migration. East Asia itself contains enormous diversity because of its size, geography, long history, and multiple population expansions. Europe also contains substantial internal diversity, but its demographic history includes strong bottlenecks and major population turnovers. These differences shape the statistical structure of modern genomes. A population's diversity is a record of its demographic history, not a measure of its biological quality. When scientists say one population is genetically more diverse than another, they are not saying one is better. Diversity can increase resilience in some circumstances, but it can also contain variants associated with disease. Low diversity does not mean biological weakness. Population genetics is about historical processes. A population may lose diversity because a small group founded it, because a population bottleneck occurred, or because certain variants were strongly selected. Another population may retain more diversity because it remained larger or experienced repeated migration. These are historical explanations, not rankings. This becomes particularly important when interpreting the genetics of disease. If a risk variant is common in one population, it may reflect adaptation to an ancient environment or simply demographic history. A disease that is more common in one population may also be influenced by diet, healthcare, pollution, occupation, social conditions, and other environmental factors. Genes are part of the explanation, not automatically the whole explanation. The strongest modern studies, therefore, combine genomic information with clinical and environmental data. The goal is to understand interactions rather than assign diseases to races. The future may make this comparison even more revealing. As researchers sequence more ancient genomes from China, Central Asia, Siberia, and Europe, they will be able to reconstruct population movements with greater precision. As modern cohorts become larger, scientists will identify rare variants that previous datasets could not detect. As algorithms improve, polygenic risk scores may become more accurate across ancestries. And as researchers integrate genetics with metabolomics, proteomics, microbiomes, and clinical records, they will begin to see how genetic variation influences entire biological systems rather than isolated genes. That is the real revolution. DNA is no longer being treated simply as a code that tells us where someone came from. It is becoming a tool for reconstructing history and understanding disease. A genome can reveal ancient migration. It can identify population bottlenecks. It can show where selection occurred. It can reveal how different populations responded to different environments. It can help explain why a medication works differently in different individuals. And it can show how two populations that appear very different on the surface remain deeply connected at the molecular level. So when a Chinese genome and a European genome are placed side by side, The answer is not a simple list of differences. It is a layered map. At one level, ancestry patterns distinguish the populations. At another, ancient DNA connects both populations to people who lived long before modern identities existed. And at the deepest level, the comparison reveals something almost philosophical. Human diversity is the product of separation and connection happening together. You are carrying the history of migrations. You are carrying adaptations to environments your ancestors may never have consciously understood. You are carrying genetic variants shaped by diet, disease, climate, and chance. And you're carrying the same fundamental biological machinery carried by every other human being alive today. That is why the most important result is not that Chinese DNA is different from European DNA. It is that the differences are understandable. They have histories, they have causes, they have consequences, and they exist inside a species that has remained connected for its entire existence. The genome does not tell us that humanity is divided into separate biological races. It tells us that populations have histories. Those histories leave statistical signatures in DNA. Some signatures are ancient, some are recent. Some affect disease, some affect metabolism, some are almost invisible, and many remain unexplained. The job of science is not to turn those signatures into a hierarchy. The job is to understand them. The 2025 cross-ancestry metabolomics research is a perfect example. More than 10,000 Han Chinese genomes were analyzed alongside data for more than 213,000 Europeans. The comparison identified hundreds of additional genetic associations and improved the precision of genetic mapping. The result was not a discovery that one population was fundamentally different from the other. It was a discovery that the two populations could help scientists understand human biology better together than either could alone. The 2025 Nature research on Han Chinese polygenic risk scores points in the same direction. Genetic prediction becomes more useful when populations that have historically been underrepresented are included. The goal is a medical system in which a person's ancestry does not determine whether a genetic risk model works accurately for them. Instead, diverse data sets allow researchers to build models that reflect real human variation. And the 2026 longevity research adds another layer. Even among older Han Chinese individuals, genetic predisposition and modifiable lifestyle factors both matter. A favorable genetic profile can help, but healthy behavior can substantially reduce mortality risk regardless of genetic background. That finding is a direct warning against genetic determinism. Your DNA is part of your story, but it is not the entire story. Perhaps that is the result that should truly shock us. We expected DNA to separate humanity. Instead, it keeps connecting us. It shows that populations diverged, but it also shows where they remained connected. It shows adaptation, but it does not create superiority. It reveals differences, but it places those differences inside a much larger structure of shared ancestry. The Chinese genome is not a completely different human blueprint. The European genome is not a separate blueprint either. They are variations of the same ancient biological system, shaped by different histories, different climates, different migrations, different diets, different diseases, different population sizes, different chances, and those forces left marks. Some marks became common, some disappeared, some became medically important. Some remain mysterious, but all of them are part of the same human story. For thousands of years, our ancestors moved across Eurasia. They crossed mountains and deserts. They followed rivers and herds. They settled agricultural valleys. They encountered strangers. They exchanged genes. Their descendants eventually became the populations we recognize today. Modern China and modern Europe may seem worlds apart, but their genetic histories reach back into a period when those worlds did not yet exist. That is the deeper lesson of comparing Chinese and European DNA. Human populations are not interchangeable because population history creates real genetic differences. But they are also not separate biological worlds. Their genomes overlap enormously, and their shared ancestry is older than the identities we use to describe them. The science is becoming more precise every year. More genomes are being sequenced. More ancient remains are being analyzed. More diverse populations are entering medical databases. More statistical tools are being developed, and every improvement has the potential to reveal another piece of the puzzle. The next major discovery may come from an ancient skeleton buried somewhere in Central Asia. It may come from a genome collected from a living population that has never been studied at sufficient scale. It may come from a rare genetic variant that scientists have overlooked for decades. Or it may come from a cross-ancestry study that discovers a biological connection nobody expected. Whatever comes next, one thing is becoming increasingly difficult to deny. Human genetic history is more complicated than the categories we inherited. Chinese and European populations are genuinely different in measurable ways, but those differences are not walls. They are footprints left by migration, adaptation, isolation, contact, and survival. And when scientists compare the genomes closely enough, those footprints begin to form a map that does not point toward two separate human stories. It points backward, toward the same ancient ancestors, toward the same migrations, toward the same long journey out of Africa and across Eurasia, and eventually toward every human being alive today. The biggest surprise is therefore not what separates Chinese DNA from European DNA. It is what survives between them: shared genes, shared biological pathways, shared ancestors, shared evolutionary history, and a future in which understanding genetic diversity may finally allow medicine to become genuinely global. The genome has no borders, it has no nationality, it does not know where Europe ends or China begins. It only records what happened. And what happened is extraordinary. We separated, we adapted, we changed, we migrated, we mixed. And somehow, after tens of thousands of years, we are still unmistakably one species. That is the real result. And perhaps the most shocking thing about it is that the story is nowhere near finished.